In our previous video, we saw the process of transcription where the double-stranded DNA was converted into the single-stranded RNA. And at the end, I mentioned this particular RNA that was produced still was not ready to move to the next stage, that is translation. So basically, this RNA has to undergo three different stages of processing, or what we call it as the post-transcriptional modification. The first stage of this RNA processing is called as Phi Dash Capping. This Phi Dash Capping occurs early in the transcription, and in this particular capping process, the enzyme involved is called as guanosyl transferase. This guanosyl transferase enzyme adds the Phi Dash methylguanosine to the Phi Dash end of the mRNA. So as the new compound is added at the Phi Dash end, which we call it as the head part, it's called as capping process. This cap is very important for the translation initiation and also for export from the nucleus. This is the diagrammatic representation of the Phi Dash capping process. In this image, you could see there are three strands. So the first two are the DNA strands, and the last red color strand, it is the newly formed mRNA. And to this mRNA, at the Phi Dash end, the guanyl transferase enzyme add the methylguanosine. So this process we call it as the Phi Dash capping. The second process of RNA is we call it as 3' poly A tailing. Why it's called as poly A tailing? Because multiple amount of A, that is adenosine, is going to be added at the tail region, which we call it as the 3' end. Like we call the Phi Dash end as head, that's why the process is called as capping. Similarly, the 3' end is like a tail for this sequence, so the process is called as poly A tailing. In this process, the polymerase, that is poly A polymerase, adds 150 to 200 adenine residues to the 3' end of the mRNA. This poly A tail increases the stability of the mRNA in eukaryotes, and this is very important to stabilize this mRNA strand. Why? Because if you see this strand very clearly, this completely consists of only adenine. So there is no chance of this tail to join back to itself and bind. So this is very important for the single-stranded RNA to form any double-stranded structure. Next process is called as splicing. What does the splicing means? Already we have studied the sequence of a gene in the starting of this unit. So splicing is the process where we are going to remove out the unwanted sequence, that is introns, and we are going to join the required sequence or the sequence which is going to code for the protein, that is called as exons. So to do this, we are going to utilize the help of snRNPs. So snRNPs are nothing but small nuclear ribonuclear proteins which contains RNA and proteins. Frequently, the splicing site in the intron has a G and U at the Phi Dash end and an A and G at the 3' end. The snRNPs then align these ends in a lariat formation to allow the precise splicing. The complexes usually contains snRNP, mRNA and the associated proteins, which together we call it as spliceosomes. So if you see this image, the snRNP along with the associated proteins, it's going to splice out, that is, it's going to cut out the introns, which is the non-coding regions of your RNA, and it's going to join the exon regions, which is the coding part of your mRNA. So once this Phi Dash capping, 3' tailing and the splicing is completed, your mRNA is completely ready and it's matured enough to move out of the nucleus to the cytoplasm, where it's going to proceed to the last stage of your central dogma, that is translation, from which you get your final product, that is protein.